So last day we had, I had started with this particular slide, but somehow in between the connection got disconnected and I had to end it. So let's start from here. This is basically a picture of how the T lymphocytes are activated. So the extreme left column shows the activation of T helper cells. Then we have the activation of T cytotoxic cells. And this is the last column is a picture of the generation of cytotoxic T lymphocytes and the phenomena of cytotoxicity. So let me start with here. Now, we know that the activation of T cells takes place through three basic signals. The first one is signal 1. Signal 1 is delivered when specific peptide MHC complexes displayed on a dendritic cell bind to multiple copies of T cell receptor being expressed on a naive T helper cell or a T cytotoxic cell. Now, as a result of this aggregation, what happens is that immediately a signaling cascade starts. The aggregation of the peptide MHC bound TCRs coupled with the conformational shift that occurs in the CD3 chain results in a downstream signaling cascade. Additional intracellular signaling enzymes are then recruited to the cytoplasmic tails of the CD4 or the CD8 receptor and the CD3 chains. Together, this enzyme-mediated cascade of chemical reaction leads to the final activation of several other enzymes. So when this activation cascade occurs for multiple T cell receptors, the T cell receives signal 1. So this cascade should occur together for multiple T cell receptors for the T cell to receive signal 1. Now, once the signal 1 is received, now what happens? Let's concentrate on two receptor ligand pair that is being showed here. This receptor ligand pair is between B7 and CD28. B7 is present on the surface of dendritic cells and CD28 is present on the surface of T cell. And this receptor ligand interaction is between CD40 and CD40L. So we have a receptor expression of CD40 by the dendritic cells and the CD40 ligand by the T lymphocytes. Note, when we compare the scenario of T helper cells with that of T cytotoxic cells, there is no pairing between the CD40-CD40L ligand because the cytotoxic cells do not have the CD40 receptor. So once a signal 1 has been perceived and received by the T helper cells, next what happens, the T helper cells and the dendritic cells starts upregulating the B7 and the CD28 molecules. And a very threshold level signaling starts through these two receptors. During this phenomena, simultaneously what happens, the dendritic cells now starts upregulating the transmembrane CD40 molecule, and this CD40 molecule then pairs with the CD40 ligand. Thus, when the CD40 ligand of the T helper cells engages with the CD40 expressed by the dendritic cells, it is then that the dendritic cells greatly increases its expression of B7 and thus its binding to CD28 on the T helper cells. So the ligand receptor interaction of the CD40-40L helps in facilitating the interactions between B7 and CD28 and further a strong signaling starts emanating from this pair. So thus the vigorous signaling 2 is achieved. Now if we look into the scenario of the T cytotoxic cells at that given moment, as I said, there is no CD40 ligand for T cytotoxic cells. So once a signal 1 is received by T cytotoxic cells, the cytotoxic cells do not have any mechanism by which they can bind with the dendritic cells to upregulate the B7 molecules on the dendritic cells. So what does the cytotoxic cell do? There's another strategy. What happens once these dendritic cells are properly activated by the, or rather once the dendritic cell T helper cells gets properly paired and the signal starts occurring through these pairs, it is said that the dendritic cells are now have been licensed, given the fact that they have their CD40 receptors upregulated. So it is this licensed dendritic cells who already had a previous engagement exposure with the T helper cells can now interact with the T cytotoxic cells. So immature dendritic cells which are not licensed are not capable of interacting directly with the T cytotoxic cells. It is only the licensed dendritic cells who have already been activated by a previous engagement with the T helper cells can actually interact with the T cytotoxic cell to produce the second signal in a T cytotoxic cell. So the B7 CD28 signaling occurring in a heightened manner is the perception and the receiving of the second signal, signal 2 by the T helper cells. Now, as I said, the CD28 signaling is very important. So co-stimulation via CD28 has some important molecular effects. One, the interleukin-2 receptor expression is induced on the T cell surface, allowing the cell to receive signal 3. So unless signal 2 is perceived, signal 3 doesn't occur. Second function of this co-stimulation is that the T helper cells start to secrete large quantities of interleukin-2 as well as other important cytokines and chemokines. The third function, the expression or upregulation of additional co-stimulatory and regulatory molecules occur in both T helper and T cytotoxic cells. And the fourth reaction of the fourth function of this co-stimulation is that intracellular signaling supporting T cell survival, proliferation, metabolism is promoted. So once the co-stimulation happens, the signal 3 is received and we get the effector population of T helper cells. Once the T cytotoxic cells interact with the licensed dendritic cells, they also receive the signal 3 and we again here get the effector population of cytotoxic T lymphocytes. So thus, once the signal 3 is received, now we are getting a functional effector population of both T helper and T cytotoxic cells. So now coming to the T cytotoxic cells and its function, we saw the activation of the T cytotoxic cells to the cytotoxic T lymphocytes. So if we look at the cytotoxic T cell responses, it occurs through 5 stages. The first stage is the activation of a naive T cytotoxic cell by a licensed dendritic cell in a secondary lymphoid tissue. Next, the proliferation and differentiation of the T cytotoxic cells into daughter cells called pre-cytotoxic lymphocytes. So this is the activation of the first stage. Second stage, it is the proliferation and differentiation into pre-cytotoxic T lymphocytes. Now, the differentiation of the pre-cytotoxic T lymphocytes in an inflammatory site into an armed cytotoxic T lymphocytes. So you can see the differentiating cytokines. So these are pre-cytotoxic T cells which are in the third stage activated to give rise to the armed cytotoxic T cells by the help of the cytokines IL-12, IF and gamma IL-6. Fourth stage, activation of the armed cytotoxic T lymphocytes by encounter with specific non-self peptide presented by MHC class 1 on a target cell. So you can see the armed cytotoxic T lymphocytes in the fourth stage are being activated by a target cell, and the activation interaction is occurring through the MHC class 1 molecule. Okay, MHC class 1 peptide molecule, and these are the co-stimulatory receptors. And finally, the cytotoxic T lymphocyte mediated destruction of the target cell occurs through the release of certain molecules from the cytotoxic T cell, or it might opt for some other pathways which we'll be discussing in the next slide. So keep in mind that from a naive T cell to a cytotoxic T lymphocyte, we need the presence of a licensed dendritic cell for activation. Once we get pre-cytotoxic T lymphocytes, a second set of activation is occurring to convert it to an armed cytotoxic T lymphocytes. It is once that these cytotoxic T lymphocytes are armed, then they only bind with the target cell, which is exhibiting a non-self peptide or an aberrant peptide via class 1, and then kills its target cell.